been here for a long time now, just studying bugs and working in the collection. So speaking of collections, you can see behind me here these big cabinets and all that. So right on the other side of the wall, in front of me here, you guys can't see it, is the big University of Guelph insect collection. So this is where we keep all the specimens. So it's like a big natural history museum, because we don't have all the displays like you maybe see at the Royal Saskatchewan Museum or anything else. We have all the specimens. They're all kept in cabinets like this, and there's about 3 million in the room that I'm sitting in right now. 3 million insects from all over the world. And we're actually Canada's oldest collection. So some of the older specimens that we have in the collection are actually older than Canada is. So Canada's 150 years old today, or this year, right? Big celebrations all over the country. We were actually founded in 1863, so we were actually a few years older than Canada is, which is kind of cool. It gives us a lot of ability to look back in time, like a time machine, and look at the insects that were present, a long time ago, and see how those insects have changed between now and then. Which species are here, which species aren't here anymore, how have they moved around, and all that. So that's basically what I do. I grew up in a rural community. I worked on dairy farms and all that as a kid, and now I'm doing the science thing here. So, do you guys have any questions, kind of about taxonomy, about insects, or anything like that, or even about going to university for biology? You guys are seniors, right? Like, this is your final year of high school. You're all getting ready to move off and do other cool things next year, right?
Yeah.
Yeah, that's cool. You guys thinking of going to university for biology?
I'm lab tech.
So, lab tech over there. Some of them are a little bit unsure. Nursing, potentially. Teaching. Trades. Engineering. Yeah, so kind of a diversity here.
Oh, that's good. That's good. One thing that piece of advice that I'd give to you guys, especially if you're interested in fields in biology or maybe one day becoming a scientist, you never know how things may change. But don't just take the biology courses. I know they're the most fun and they provide you a lot of context and are really interesting. But make sure you take a lot of English courses and writing courses as well. It's one thing I regretted when I was doing my undergrad, and now looking back is being able to write and communicate what it is that you're studying and understanding and trying to discover is a really important skill. So I know it's easy to get past your core English requirements when you're like, I don't want to do that anymore. That's annoying. Give me all the biology I can get. But it's important to keep up with the English as much as it pains me to say that. So that would be some advice I've got there for that. So a lot of people sometimes think that taxonomy isn't a field that's still relevant. But the truth of the matter is that there are so many species left to be discovered all over the world, including here in Canada. So looking just at insects, actually, we're guessing that there's about 70,000 species of arthropods, so spiders, insects, crustaceans that live on land or in freshwater environments, those sorts of things, millipedes, centipedes. We're thinking 70,000. That's a roughly estimate of how many we think occur in Canada. And to date, we've only documented about 40,000 of those. So some of those things are things that live here that have been described from, say, the U.S. or from Europe that we just haven't found yet, haven't recognized as actually occurring here. But a lot of those are actually new species that nobody has given a name to, or in some cases may never have actually seen or recognized either. So you don't have to travel all over the world to find new species. You can find them literally in your own backyard. Actually, interesting story. There's a story coming out of Saskatchewan just right now where there's a new pest in canola that didn't match up really well with what some of the agricultural communities were looking at. It wasn't working. Their tests for some other type of fly weren't working. And I just assumed that this was some weird variant of the fly, but just research that came out this last week is showing that actually there is a new species of fly in the canola fields that's causing different damage. It looks very similar to the old species that has been researched for a long time, but now agricultural researchers in Saskatchewan have identified it as being brand new, needs a name, and needs an entirely different easy to deal with in canola fields. So there's a case right from around you guys actually. But if you want, I can show you a species that I'm working on that's from my backyard. See if I can screencast here. Did it before. How do I do it again? Let's see if I can figure it out. So you guys are just learning about taxonomy now, right?
Yes.
Yeah.
Yeah, we've just started some basic taxonomy and the kingdom class phylum things. So very, very introductory right now.
Awesome. So I should clarify that I work mostly at the genus and species level. A lot of the higher stuff, the kingdom, phylum, and all that stuff hasn't changed a whole lot. We figured that stuff out pretty early, and that stuff's not changing. So where a lot of the work in taxonomy is going on right now is that genus and species level. Some at the family level as well, figuring out how different genera are related to one another and where to start drawing those lines between what the genus, what the family, and where all those things should go. Let's see. I know I can do it. I've done it before. I just can't find the thing. Sorry, guys. I don't know. Maybe I won't be able to do this like I hoped. Oh, there it is. Alright, can you guys see?
Yes, we can see your computer.
Okay, cool. I'm going to bring that up because I can't see it right now. Okay, so these are some of the slides that I work on. These are the slides that I work on for the core of my research. These are called stilt-legged flies for obvious reasons. They got really long legs. They're in the genus Micropisa. And so I'm working on these. Like I said, most of the ones that I'm working on are in Central and South America. And so when I'm studying them, I'm looking at a lot of dead specimens, so specimens that are kept on pins like these. And this is what is all in the cabinet behind me. Is a whole bunch of specimens preserved on pins like this from all over the world and from altered times. And so what I'm doing is I'm looking at them very closely and I'm trying to look at all the different characters. So this is actually a female. This is the ovipositor. This is the thing that she uses to deposit her eggs with. This one's really weird because it's got a couple of prongs down at the end, which are totally unique. No other species that I've ever seen has got these appendages on the end, and I have no idea what they could possibly be for. So it's good for identifying the specimen and knowing that this species is one thing, but it leaves more questions. Like, what is this species doing out in the wild? Why does the female need these crazy arms on the ends of their abdomen here? To help them dig into something? Are they using it to help with courtship? Are they using this for mating and all that other stuff? No idea. So that's the cool thing about taxonomy is that as you're going through all these specimens from museums and collections all over the world, you start finding all of these really weird, unique things that bring up more questions that you want to answer through field work and through other research studies. But I work with a lot of different characters, so I'll be looking at the arista, so how different parts of the head are different between species. I'll look at all the bristles and stuff like that and tell, are they in different places? So there's a lot of fine detail work in what I do and saying, is this the same as that? Is this different? And so the other thing that I do is a lot of comparative work. So these are male genitalia from a species of fruit fly that I'm currently working on. This is something that's known from Ontario. So we've got two different things. So there's very slight differences between these two things. So that's basically what my whole job is looking at one thing and then looking at another and trying to tell what the differences are between them. So do you guys see any differences between these two photos?
What do you notice?
Well, that one's bigger than that one. The one in the
Okay,
top.
so some size difference. What else do you notice?
That one seems to be a bit of a different shape.
Okay.
The one on the
Sorry,
right.
what was that?
That one seems to be a bit of a different shape than that one. That one's more curved, but that one is a little more rough.
Are you able to hear her?
I missed a little bit. Could you repeat it? Just because
Come on up here so that you can get closer to the speaker set.
Okay. One I'm just trying to think. The left one seems like it's rounder, while the right one seems like it's a little more rough in shape. That's one of the things I noticed. Anyway.
Yeah, that's good. So you guys are on the right track. This is basically what I spend my day doing, is looking at stuff. So some of the characteristics that I'm looking at, you see how the ones on the right here, they're kind of splayed out, so the tips are further apart than the bases are, versus the one on the left, they're sort of parallel or somewhat even facing into one another. So those sort of minute little differences, you guys are good for looking at the shape of the top parts there. Even how these little barbs are overlapping with one another. These ones are kind of, the one on the right here. For the photo on the left, the barb on the right is considerably higher than the one on the left, whereas the other ones are a little bit more overlapping. So that's basically what I spend my day doing every day is looking at this and comparing them. And then it gets more complicated as you add more species in, and then you have to look at all sorts of other aspects of the morphology and trying to figure everything out. But here's what that species looks like. So this is the new species. This was something that we had overlooked. We had thought that it belonged to another species until we did some genetic work and we sequenced its DNA, and somebody else actually came along and compared the DNA from specimens that they had collected in their area over in Michigan. And they didn't match. And so they contacted us and were like, something's going on here. We don't know whether this is accurate or not. Can we look into it more? And as it turns out, this is a different species from this. So these are the two sister species, what they look like on the outside. There's not a whole lot of differences to be seen on the outside. And so we're looking for all these minute changes in their genitalia, trying to look at. So this is the male genitalia, all of it. The relevant bits, this is what we call the Aedeagus, which is basically the fly's penis. This is what we call the sperm pump. So this is what fly junk looks like, basically. Welcome to biology class. And so we're trying to look for a whole bunch of little differences between this and this other species here. And so we'll do the same thing here, and we'll look at female genitalia, we'll look at the outsides, we'll do as much as we can, try and see as much as we can. And we'll rely on DNA as well to help give us differences between them. So it's a lot of back and forth and comparing different things to see how they work.
Do you do a lot of DNA work?
That's just what it is today.
Do you do a lot of DNA work?
I do some. I don't do a whole lot because we have facilities here. So I work with a team up at Agriculture and Agri-Food Canada that does a lot of that stuff. And so I'll go up there. Even one of the people there is my advisor, and so we'll go up every once in a while and we'll sequence some DNA from specimens. And we'll ask a bunch of different questions, whether these two specimens are the same species or whether these two species belong closely related or whatnot. But a lot of my work is also based on morphology, so just using a microscope and looking through and trying to do that. Is this the same as A versus B, A versus C, B versus C, that sort of thing.
Okay.
Yeah. So do you guys have any other questions, or should I leave you to go, or anything else that you're interested in about insects, if you've got anything going on?
I think you answered one of their key questions, which was what kind of things do you get to do during your day or your week?
Yeah. Well, there's also a lot of email. We get a lot of reading, a lot of email, a lot of writing, and then when all those things are kind of out of the way, then I get to look at bugs under the microscope. So it's a lot of balancing all these different aspects and trying to get as much going on as I can. So even I haven't even got to do the field work with my degree, but that's the best part, is getting to go travel and go to different places and collect insects at all sorts of different places and see all sorts of different things and see them in their natural environment. So
how
that
long,
doesn't happen often, but it's fun.
How long do you think it's going to take to finish your PhD?
Well, I'm hoping to be done this summer. I started in 2012, so if I finish in the summer, that'll be 5 full years for this one degree. I started university 14 years ago, and this will be my 3rd degree. So I've been here at the university for 14 years now, and I would like to not be here for much longer.
Okay. So what do you hope to do when you're finished?
That's a really good question, and I don't know. I'm trying to keep as many doors open for what I can do afterwards. The standard track is I would go off and be a professor, hopefully, and teach other students and do research and communicate all those things as well as I can. But it's tough to do that. There's not as many jobs as there were previously or as there have been in the past. And so I'm also keeping my options open. I'd love to work in a museum, Natural History Museum, like the Royal Saskatchewan Museum would be great, or the Canadian Museum of Nature would be awesome. Basically doing the same thing that I'm doing now. I could also maybe be working for the government, working as a staff scientist, helping to understand the agricultural importance of insects and the taxonomy and systematics of those insects that are relevant to Canada. So I have a bunch of different options. And again, I've developed a bunch of skills besides just knowing which end of the insect to look at, for that I can apply to various other aspects as well, professional writing, that sort of thing. So I'm keeping as many doors open as possible. I don't have anything to do after this, but there's a lot of different ways that I can use my knowledge and my skills down the road.
Do you have some other questions?
Yeah, so one thing I wanted to point out is if you guys are interested in it and learning more, the Royal Saskatchewan Museum has got a great collection there, and they've got a really nice entomologist named Corey Silas. He works on bees primarily, and he's a taxonomist, and he works in their collection there. And I bet he'd be more than happy to talk to you guys if you're interested in learning more about insects or interested in learning about taxonomy, or as you go off to university, perhaps he may be somebody who could provide you some internship opportunities if this is something you're more interested in going into.
I think some of the boys and I were wondering, why did you pick flies?
Why did I pick flies? That's a good question. So primarily I picked flies because that's what my professor studied. So I kind of inherited that from him because that's his main research focus. But also when I was taking my introductory entomology course, the insect course, that kind of changed what I wanted to do. That kind of gave me the idea that insects is where I wanted to be. As we were going through all the different insects, seeing beetles and grasshoppers and all that stuff, it was the flies that really spoke to me. That just kind of captivated my interest in my imagination, and that's basically why I stuck with flies, just because I thought they were cool. There was so much we still don't know about them, and that very few people are working on the flies. A lot of people work on like butterflies and moths and beetles. Those are all super popular, but not as many people work on flies. So I figured it might be a better opportunity to fill a niche within the academic world as well. So we'll see if that strategy pays off. But I've loved it. It's been really, really interesting to see the crazy diversity of flies and see all the different things that they do. They're by far the most diverse in their roles within the insect world and within our biology.
Do you have other questions? They're not usually this quiet, honestly.
I know, they just didn't all the insects that are around them that they don't know about. The ones crawling across the back of the room right now. But for sure, there'll be insects crawling around under your desk right now without you even realizing it. So that's how insidious they are. It's great. Awesome. Well, I'll let you guys get back to your class then. Hopefully you enjoy it. If you guys have questions, you can always reach out to me. I can give your teacher my email. I'm on Twitter as well, if you guys are on Twitter. That's my own focus. If you guys have questions or photos of insects that you want help identifying, I'm more than happy to help you with that. And yeah, hope you enjoy the insects that are around you. It's the easy part to see and feel. But you don't have to go to exotic places to find really cool things.
Thank you so much for Skyping in with us. It's been great to hear about your work.
No problem. Anytime.
Have a wonderful day. Keep warm.
Yeah, thanks. Not a problem here. Hope you guys do as well.
Thank you. Bye-bye.
Thanks.